Hello and welcome to the Cheeky Science Show. So the last time I spoke about the coronavirus, I said this. I said to myself that I wasn't going to do another video on the coronavirus because mainly reading all the news pretty much terrifies me. And so I'm still pretty terrified, but on this channel I like to talk about science, and there's a lot of really cool science and research coming out on the coronavirus. And so one particular article caught my eye, and so we're going to talk about it in this video. And so what this article was trying to address was why certain populations, such as the elderly, are more vulnerable to coronavirus infection, and it relates this vulnerability to a mechano-genomic regulation. So in this video we'll talk all about that and my interpretation of this article, which actually isn't really an article, it's a comment, so there's no recent data, but it discusses previous data and comes up with its own hypothesis to explain this. But firstly, let's just describe what the coronavirus is. So coronaviruses belong to a family of positive strand RNA viruses, and through infection of their hosts they can cause upper respiratory tract illnesses such as ARDS, which is the acute respiratory distress syndrome. And so SARS-CoV-2 is the coronavirus that is currently causing this current pandemic of the COVID-19. And so what we've unfortunately witnessed in this most recent outbreak is that coronaviruses are potentially highly infectious. And not only is it highly infectious, but it also can have considerable fatality rates, as we've also evidently seen. However, these fatality rates are particularly high in high-risk groups that include the elderly and others with underlying chronic medical conditions. And this has a huge impact on the economy and poses a really great burden on the healthcare services. But the question is why? Why do they have such a high risk of infection and then fatality? So currently the main hypothesis for why this would be the case is that these high risk groups have a dysfunction of the immune system, which makes them more susceptible to infection, and then their ability to deal with the situation. But what was described in this comment article is an alternative hypothesis, or one that could also go in combination, which is an altered cellular mechanical state. And so what this refers to is the differences in the cell's response to their surrounding environment. So to give an analogy, let's say I'm surrounded by two sponges that are moving towards me. Maybe not too bad a situation, but put two concrete walls besides me that are moving closer together and I might not be too happy. So you see my response has changed because the environment is different. And as strange as that analogy probably was, the same can be said for cells. Cells reside in tissue and they're surrounded by other cells, but they're also surrounded by an extracellular matrix. And cells need to be able to interact with and respond to changes in its environment to be able to survive, and to help prevent the cell from going rogue and causing cancers. So the point I'm trying to make is that cells effectively talk to their environment and their surroundings, but not only do they do this by chemical signals, but they can also interact and communicate through mechanical signals. And it's the conversion of mechanical signals to biochemical signals that can propagate within the cell is referred to as mechanotransduction. So I'm planning on doing a future video where I go into a bit more detail about mechanotransduction because it is really interesting, and I've read a lot about it recently regarding an overlap with my PhD project. But for now I'll just give a kind of generic overview to what really is mechanotransduction and what is meant by mechano-genomic regulation. So what I'm drawing out here is the extracellular matrix, which I said is what surrounds these cells. And so then I've drawn out the cell membrane. And so this is the barrier to the cell. And then what I've drawn on the bottom here is the cell nucleus, which contains the DNA that contains the different genes that are regulated and expressed within a cell. And so what you can see is at the membrane there are certain receptor proteins that can actually interact with the ECM, the extracellular matrix. And through a mechanism that I won't describe here, it can also interact through to the nucleus and transmit signals that can alter the gene expression patterns. And so these gene expression changes, the cell's responses, can differ depending on the stiffness of the ECM, such as in my analogy between the sponge and concrete. So what does this have to do with the coronavirus? Well, if we think about what the coronavirus wants to do, it's programmed to make more copies of itself. and to make more copies of itself, it exploits its host, and therefore it exploits the machinery that's in the human cells. But critical to its success, it wants to do this such that it's undetected. And so previous work, and I just want to point out here that the references I've used, I've put the links to the papers in the description box, have emphasised the use of the NF-kappa B pathway in this process. So what is the NF-kappa B process? Well, NF-kappa B is a protein, and it does a lot of things in the cell, but one thing that it does is it can promote an inflammatory response. And so this response can bring in the immune cells that can then destroy and kill infected cells. But studies have also shown that this protein and its response can also be activated by the coronavirus to promote its replication. But how would this work if the coronavirus wants to remain undetected? Well, as I said, the NF-kappa B pathway does a variety of things, and so what is now thought is that the coronavirus actually fine-tunes this pathway. And so this means that it can still replicate, but still try and remain undetected. So what has this got to do with age? Well, the coronavirus largely infects the lungs, and the respiratory system in aging individuals is characterised by fibroblast dysfunction. This can result in increased deposition of the extracellular matrix and increase the tissue stiffness. And going back to what we said earlier, cells respond differently when they're witness to different stiffnesses in their surrounding environment. And one pathway that can be altered through a different stiffness of tissue is the NF-kappa B pathway. So does the virus exploit this such that it can infect cells that are in a stiff environment? So if you take some cells that are growing on either a soft or a stiff matrix and you expose them both to the same inflammatory signal, you see that they both activate the NF-kappa B pathway, but they both have differences in the downstream genes from the NF-kappa B that are expressed. And so just imagine that those different genes are currently within that purple circle. Now, the interesting part is that if you also take a list of genes that are differently expressed by coronavirus infection, there's a substantial overlap between these genes. So genes that are expressed differently depending on the mechanical states, i.e. if it's soft or stiff, could have some interaction on impact on the coronavirus infection. And so this overlap is actually quite large, and therefore it's interesting to look at these genes that are affected by both of them and to see if we can learn anything from that. So it seems that most of these genes have important immunomodulatory effects, and so it could be that the coronavirus can exploit the vulnerability of cells in a stiffer environment and improve their chances of replication without the cells knowing that they're there. So as I said, this is currently just a hypothesis, but I think it could have important implications not only for the coronavirus but for understanding other viral infections. So I hope you learned something, and as always, thanks for listening.